So our next module that follows with chapter 4 in your book is a description of genetic control of your cell function and some of the basics of inheritance patterns. And a lot of the information in this chapter and in this presentation will sort of inform our next module, which is what happens when we have genetic and congenital disorders. So this is a bit of a lead in into some of the things we're going to talk about. The other thing I'm going to mention here too is that this is a very simplified version from what is in your reading. I took a lot of kind of things out just to try to really hit the highlights of the stuff that I think will be important for you to know going forward. Some of you, based on maybe what your background is, as far as previous bachelor's degrees, may have a more advanced kind of knowledge of things like genetics, that kind of thing, than what you see here, and that's great. But if you don't, and this is your first introduction, I think, taking what we talk about in this presentation plus, weeding through some of the stuff in the book that's closely related will be plenty for your understanding of this topic going forward. Okay, so our first unit learning outcome is really to kind of compare and contrast DNA versus RNA. So I tried to set this up so you could see sort of some of the differences, and I picked, some of similar things to show you what was different between the two. They're both involved in, genetic replication, but DNA, as far as its structure, is double-stranded. One of the features of being double-stranded for DNA is that it gives it a high level of structure and stability. And that structure and stability is important for DNA because you're finding that DNA in the nucleus of every single cell throughout the body. And it's replicating itself kind of over and over and over. And it has to withstand, kind of a lot of environmental influence, and keep itself kind of within that structure to be able to continue to replicate. So that stability of that double strand is really important to kind of maintaining the physical structure of DNA itself. Again, it's found in every cell within the nucleus. We're going to talk a little bit about that later because I think that's an interesting thing to think about, right? That every single cell in your body has really the same kind of DNA. So why do cells behave differently, right? So we'll talk about that a little bit on a slide a little bit later. So DNA contains all of our genetic information. RNA, on the other hand, its primary responsibility is to synthesize proteins. And so we look at our genetic material and we think about, well, our genetic material and our DNA, that's really what makes us who we are. But the reality of it is, it's those proteins that are created eventually by RNA that are really what runs the show. They're what, create our biological functions, right? Like the proteins are really what's doing the heavy lifting, and those proteins are synthesized by the RNA. That RNA is directly transcribed from the DNA. We'll talk a little bit about that, and that happens within the nucleus. And then it's carried to the cytoplasm of the cell where it's translated into proteins. So we're talking a little bit more about that protein synthesis process. So mRNA, or messenger RNA, is like the roadmap for protein production. And in this first little picture up here, you can see how there's our double-stranded DNA helix, and it's being transcribed within the nucleus of the cell into RNA that then is turned into this messenger RNA, again, our template for protein synthesis created during transcription. And then that messenger RNA is going to move out of the nucleus into the cytoplasm of the cell. This second picture here is really where those proteins are being made. We call that translation. So this is happening on the ribosomes within the cell. So that's another organelle of the cell, if you recall that from cellular biology. And that's really where the reading of those instructions coming from the mRNA is taking place. So that ribosomal RNA is really involving the physical structure in the cytoplasm where that protein synthesis takes place. Okay. And what you see happening here on this ribosomal RNA is that another form of RNA called transfer RNA, that's our little friend kind of right here, is carrying these little individual amino acids and matching them up to that roadmap that sits on my RNA strand that was, again, ultimately transcribed from my DNA. And each of those amino acids get attached to that ribosomal RNA by our transfer RNA. All proteins are made up of amino acids. They're joined end to end to form one of these long polypeptide chains of protein molecules. So again, that transfer RNA is delivering those activated amino acids to the protein molecules right inside the ribosomes. And that's what ultimately carries us all the way from double-stranded DNA to that synthesis of protein in the end. So as we mentioned, we have DNA in every cell of the body, but only about 2% of the genome encodes instructions for the synthesis of proteins, like you just saw. The rest of it are non-coding regions, they're structural. They help to determine where, when, and in what quantity proteins are made. And so what kind of proteins we're making through that process differs depending on what what the function of those cells are, where we are in the body. They all have to kind of do all those different things that are necessary, and they don't all do the same things, right? So something has to be telling us what kind of proteins are we going to make and what kind of proteins are we not going to make. So the degree to which a gene or a particular group of genes are actively being transcribed into proteins, that's called gene expression. So gene expression, the degree to which a gene or a particular group of genes is active. Okay? Transcription factors are a special kind of protein that function to either increase the activity of transcription or decrease it, depending on what kind of substances we need to make, right? And when we say we're going to increase gene expression, we call that induction. And when we are going to repress or turn off the expression of a gene, we call that gene repression. That's a process by which a regulatory gene acts to reduce or prevent gene expression or that production of protein. Okay. So there are activator and receptor sites commonly monitoring levels of the different synthesized products throughout the body, and they regulate that gene transcription through a negative feedback mechanism, meaning that as the amount of a certain product of gene expression increases, that negative feedback turns off further gene expression. It's kind of like the thermostat in your house, right? If you set your thermostat at a certain level, mine is set at 68 degrees. If my heat turns on, it turns on when the temperature is below 68. The minute the temperature hits 68, that signal is fed back to the furnace to say, you know what, it's warm enough, let's turn the heat off. Same thing in genes. Again, if you have enough of a certain substance that's being produced by the expression of that gene, you have some kind of activator, well, actually in this case, repressor site that says, okay, we have enough, and it turns off that gene expression. Okay, same thing if you don't have enough. Not having enough of a certain substance might activate those genes to create more of that substance. Okay. So chromosomes themselves, and some of the basics of chromosome composition that you should know is that there are 23 pairs of chromosomes normally within the human body. Okay. 22 of them are what we term autosomes, 22 pair of chromosomes that are alike each other in both males and females. And so you kind of can see them here. All of these kind of laid out on this map, these pairs of chromosomes, 22 of them as autosomes. And then we also have our sex chromosomes, and those are the 23rd pair of chromosomes in the body. And those determine the sex of a person. So all males have an X and a Y chromosome, and all females have two X chromosomes. So male XY, female XX. Okay. And these, I think, down here in the little corner are your sex chromosomes. All right. So how do we get that cell replication, that replication of DNA that gives us those autosomes or sex chromosomes. So there are two processes by which the cells divide and replicate their DNA material. One is mitosis. So mitosis is the replication of DNA to duplicate DNA within somatic cells in the body. And this is represented by what we think about when we think about the basic cell cycle. We start with one cell, the DNA splits and completely replicates itself, and then creates two resulting cells that should have identical pairs of those 23 chromosomes. So all the DNA material exactly splits and replicates in that mitosis cycle. Meiosis results in the formation of gametes, or what we call reproductive cells. So within reproductive cells, so these are things like sperm and ova, each cell has only a single set of those 23 chromosomes. So instead of those chromosomes being paired, as they are in our autosomal cells, we get just one set of each chromosome in those gametes. Okay. Yeah, and so then what we get in the end are what we call germ cells with just one set of chromosomal material. Okay, and that's how, in the end, you get half of your genetic material from your mother and half from your father, right? Because it comes from those split germ cells that come together. All right. So again, germ cells are those found in sperm and ova, those reproductive cells. So that's important to remember because how we think about DNA and its mutations and how we inherit certain things is kind of hinged on where different DNA mutations might take place. So DNA mutations are substitutions of one or more base pairs, and base pairs are the nucleotides that make up DNA themselves. So when a mutation happens, one of the base pairs that's supposed to be within that DNA strand in a certain order gets swapped out or deleted, or something gets added in, something gets rearranged. There's some change that happens within the DNA itself. We call that a mutation. And some of those mutations just kind of happen spontaneously, meaning there's nothing that really brings them on. Again, DNA is doing a lot of work and all its splitting and replication. And so mutations are going to happen regardless of any outside influence, just these kind of spontaneous mutations. But there are also mutations that happen because of some environmental agent. And when we talk about later, when we start talking about things like neoplasias and some of the other kind of DNA-based diseases, a lot of these things are brought about by DNA mutations that are happening by way of some kind of environmental influence, things like chemicals. So really common when we think about here is the influence of chemicals that are found in things like cigarette smoke on DNA. Radiation is a big environmental factor that can really impact the DNA itself and cause significant and often widespread mutations, especially if that radiation is applied either in high doses or at very critical times. So that's the kind of the difference there between spontaneous and environmentally induced DNA mutations. And you can see mutations in both somatic cells and in germ cells. So, we get mutations in our somatic cells all the time, and those are things that can cause problems within our own individual body, right? Over time with those genetic mutations, they can lead to things like disease, cancer formation, whatever it may be. But if you see a mutation that happens in someone's germ cells, now there's a mutation that ends up in a sperm and an ova, and that's then passed to offspring, then you can get a inherited mutation. So germ cell mutations can be inherited. So if you have a mutation within an egg or within an ova, and that ova forms, a new child, and there's a mutation in that germ cell, that mutation can then be inherited into the child. And so that kind of piece of information will become important when we talk about maybe some of the childhood cancers, because oftentimes when cancers arise in really young children, a lot of them originate in mutations that were in the germ cell. So they were just inherited by that child, right? Very different than what you see maybe in an adult cancer where you have mutations happening in those somatic cells over time. These just came right along with those germ cells. So a little bit more about some of the genetic information within the cells. So some terminology it's important to understand. One is the genotype. So a person's genotype is the genetic information stored in those sequences of base pairs found in that DNA double helix. It's what the genes say. The phenotype is the traits that you see outwardly on a person. So, phenotype, these are our recognizable traits, physical, biochemical. They are associated with specific genotypes, but we often see variations in phenotypes based on genotypes. We'll talk about that a little bit later. So, more than one genotype may have the same phenotype. So, you can have outward characteristics that look the same on the outside but have different genotypes that created them. So like some brown-eyed people are carriers of the coding for blue eyes and other brown-eyed people are not. And so it just depends on which gene was able to sort of express itself, right? What's happening underneath might be very different, but that phenotype is that trait you see outwardly. So phenotypically, two types of brown-eyed people, they look the same, brown eyes, but they genotype differently, right? So how does that happen? So other kind of like things to think about, some terminology pieces and how um, genetic information is transmitted. So the position of a gene on the chromosome itself is called a locus, and alternate forms of a gene at the same locus are called alleles. Okay. So when only one pair of genes is involved in the transmission of information, the term single gene trait is used, and single gene traits follow Mendelian laws of inheritance that we'll talk about. So again, um, alleles are alternate forms of a gene, one from each parent, and a locus is the position on the genes that are position genes occupy on a chromosome. Okay. So we have kind of two different, what we would term patterns of inheritance. So polygenic and multifactorial. So polygenic inheritance involves multiple genes at different loci, with each gene exerting a small effect on determining a trait. Multiple pairs of genes, many with alternate codes, determine most human traits. So most human traits are not coded by just one gene. Usually there are multiple genes that are coding for a human trait. Polygenic traits tend to be predictable, but less so than a trait that's being passed by a single gene. So single gene traits are the most predictable in how they'll pass from parent to offspring. Polygenic traits are somewhat predictable, but not as predictable. And then we have multifactorial inheritance. And that's similar to polygenic inheritance in that you have multiple alleles at different loci that are affecting that outcome. But the difference is that in multifactorial inheritance, we also now add in different environmental effects on those genes. So sometimes people have the polygenic coding for maybe a certain disease, right? So I'm going to use the example of maybe diabetes. There's a certain amount of inheritance that comes along with diabetes. But just because someone has a genetic coding for diabetes or genes related to diabetes doesn't mean that someone will display that physical or biological outward trait of diabetes. But if there is some kind of, the thinking is if there's some kind of environmental exposure or impact, whether it's a virus, we're just really not 100% sure what, that now induces those genes to express those traits, now we have that outward expression of diabetes. And that's kind of what we mean by multifactorial. The genetic kind of composition is there, and now we've got an environmental influence that makes that trait kind of express itself. And another person may have the underlying genes to express a disease state, something like that, but may never develop it because the environmental influence just wasn't there. So that's what we talk about when we talk about single gene versus polygenic versus multifactorial inheritance patterns. So I mentioned the term Mendelian genetics, and this is again really talking about kind of some single gene inheritance traits. And so Gregor Mendel, way back in 1865, discovered some basic patterns of inheritance. He is responsible for what we know about those terms dominant versus recessive factors within genes. He discovered that observable traits of single gene inheritance are inherited by the offspring from the parent. And he's also the person, if you're familiar with the use of something called a Punnett square, that's where this came from. I think I do have one we can look at here. So here's my little Punnett square. So the Punnett square shows all the possible combinations of transmission of a single gene trait. So the example that we'll use, because it's the one that comes from the book, is dimpled cheeks. So if you have dimples in your cheeks, those are genetically inherited, and we can track those via a Punnett square. So this example is shown when both parents are what we call heterozygous for the trait. So just kind of going back to Mendelian laws, the first law of Mendel's first law is that during maturation, the germ cells of both parents undergo meiosis and they dividing the number of chromosomes in half, right? And so we talked about that already. We have meiosis, and so now we have germ cells that have half of the genetic material from each parent, right? And so then two alleles, two sites on those chromosomes themselves from a gene locus have now separated into each germ cell, and each one receives one allele from each parent. So that's the first thing that's important to know and to track Mendel's laws, right? So you can kind of see that here, right? We have the female and the male, and both are big D, little d. They're both what we call heterozygous. So they have two types of alleles for that trait. And so when those chromosomes split, for the father, we make two germ cells, one with big D, one with little d. And for the mother, that cell splits and we get the same thing, a ova with one big D, one little d, right? So that's what we mean when we see that they separate and those germ cells retain one allele for each pair, okay? And the second law is that those alleles from the different gene loci segregate independently and recombine randomly in the zygote. So how those little germ cells, recombine themselves will be very much random, right, in the offspring. But with just these two kind of codings for traits, just the single gene loci, we can map out the possible combinations that we might see in the offspring. Okay, and that's done here. Okay. So again, in this example, both parents are heterozygous, so one big D, one little d for a trait, and the alleles carried by the mother, those are on the left, and by the father on the top. And that D allele is dominant, the big D, and the little d allele is what we call recessive. So um, big D, and big D, little d offspring will have dimples because that's my dominant trait. And the small d offspring does not, right? So if we're thinking about genotype versus phenotype, all of these genotypes, this one, big D, big D, big D, little d, big D, little d, these are all going to code for the phenotype of dimples because, again, big D is dominant, and so that is what will be expressed. If offspring gets two little d's, then this child will not have the phenotype of dimples. Okay. So again, we use these terms quite a bit already. Maybe I should have rearranged and show these first, but it's just to reiterate. So homozygotes are people in whom the two alleles of a given pair are the same. So AA or little a. And in the example I gave, neither one of those parents were homozygotes, but some of the offspring were, right? Specifically, that offspring who did not show the phenotype for dimples, they were small d, small d. That's a homozygote gene expression with two recessive genes. Heterozygotes, like you saw in my example, big D, little d, are people having different alleles at a gene locus. Okay. So again, people in whom the two alleles of a given pair are the same, big A, little a, are called homozygotes. And heterozygotes have different alleles, big A, little a, at a gene locus. A recessive trait is one expressed as a phenotype only in homozygous pairings. Okay, so the only time that recessive traits are expressed or shown as outward phenotypes are when you have a homozygous pair for both recessive traits. So again, back to my dimple example, if you want the phenotype of no dimples, you need little d, okay? Because that is the recessive trait. In a dominant trait, one is expressed as a phenotype in either homozygous or heterozygous parents. So if you inherit a dominant trait from either one of your parents, you will express that phenotype. So again, in the dimple example, big D, of course, phenotype, two dominant traits, right? But big D, little d, you will also express that phenotype because it's a dominant trait, okay? And the final thing to understand is this carrier piece. So for some people, those who are heterozygous for a trait, meaning they have one dominant, one recessive, if a person is heterozygous for a recessive trait, they won't, they will not manifest that trait, but they will carry it. So in the case of my dimple parents, right, they were both carriers for no dimples, right? They both were heterozygous. They carried a big D and a little d, but what they expressed were dimples, but they were carrying the trait for no dimples. And so there's a 25% chance of their offspring not having dimples at all because they still carry that recessive trait. If both parents had been big D, there's no chance of them ever having a non-dimpled child because neither one of them are carriers for that recessive trait.